Assalamu alaikum to all. Today we are going to discuss about the immobilization of plant cells. This technique, the immobilization technique, is mostly useful for the production of secondary metabolites and recombinant proteins. Now the question is here, what are secondary metabolites? Plants are able to produce primary metabolites and secondary metabolites. When the plant cell enters into the log phase, they are able to synthesize its primary metabolites. These primary metabolites are useful for their growth and development. So, the primary metabolites which are secreted by the plant are used by plants only. But the secondary metabolites are not required by the plant for its normal growth and development, or we can say the secondary metabolites are the byproduct of cell metabolism. So we can, in other words, we can say they don't serve any function to the plants. But it has been observed in many plants these secondary metabolites are used as a defense mechanism. These secondary metabolites can be used by plants as a defense mechanism, or they can be used for reproductive purposes. Like these plants are able to make colors, then the colors are get attracted by the bees. By bees we can get cross pollination. Likewise, they can be used for reproduction purpose. Even they can secrete some smells, like some aroma type they are able to secrete. The important secondary metabolites which plants are producing are flavonoids. These are broader categories: flavonoids, alkaloids, steroids, tannins, and terpenes. These are five important classes of the secondary metabolites. Secondary metabolite was Secondary metabolite production by immobilization technique has been observed in many plant species, and in India this technique has been first reviewed by the Roy and Ravi Shankar in 2002. They also said that the secondary metabolite can be produced at a large scale by using commercial biofermenters or bioreactors, as you have studied in the fermentation technology. So there we are able to scale up the process. For scale up the process, we can use many strategies to plant cells by which we will be able to get higher production of secondary metabolites. First is the if we will go for the biotransformation. If we will go for the biotransformation, what we will do, we will take a plant cell, we will insert our gene of interest. By this gene of interest, we will be able to produce large quantity of secondary metabolites. Then we can increase the cell permeability. How we will increase? We have to, whatever the secondary metabolites are produced inside the cell, it should be secreted outside the cell. Then only we will be able to extract the secondary metabolite from the environment or from the medium. So cells should be permeable to that much that whatever the secondary metabolite plant cells are producing, it should be secreted out. Even we can increase secondary metabolite production by using metabolic engineering. What we can do, we can insert gene which will be coded for specific enzyme, and this enzyme will be responsible for the overproduction of that secondary metabolite. So the immobilization technique is applicable to those plant cells which involve two important steps First is when these plants are able to increase their biomass, means their biomass production should be increased first. Then in second stage, they should be able to form the secondary metabolites. Immobilization techniques are applicable to those plants which will involve two-step strategy. In first step strategy, they have to increase their biomass production. Then for the second case, they should be able to produce secondary metabolites. Like the like the important plant for which immobilization technique has been applied is the capsicum frustescens. In this case, they are able to produce caps- capsaicin, and it has been observed that this capsaicin is produced 50 times more by using immobilization technique than the cell suspension culture. The another important component, secondary metabolite, dysgenin. Dysgenin has been improved by 40% production in immobilization technique over the cell suspension culture. By taking these cells in the polyurethane, by taking these cells entrapped in the polyurethane and medium containing 3% of sucrose. However, it has been said now that commercial production of the secondary metabolite by immobilized cells is yet to be developed, as we are not able to produce secondary metabolite for all plant species. We are having very limited plant species in which we were able to develop this technique, or this technique is not applicable to the all plant species but to few. Still, scientists are working on it, how we will improve the secondary metabolite production from the other plants. So we can, for entrapment method for immobilization of cells, we are having alginate, as we can use alginate for Arabidopsis thaliana. And the Arabidopsis thaliana is a cell which is heat sensitive. For entrapment of Arabidopsis thaliana, we can use alginate, or we can use polyacrylamide, polyurethane, or we can use agar and agarose. So now we have used the entrapment method. So what we have to do, we have to first, for the production of secondary metabolite, first we have to see the plant, which we have to choose the plant which will be able to produce high yield for your specific secondary metabolite. If it is not so, but then what we can do, we can go for the biotransformation technique, in which our gene of interest, which is responsible for the overproduction of secondary metabolites, will be inserted into the plant cell. Then we have to allow it to grow to make the whole complete plant. Then this plant can be used as your explant for the secondary metabolite production. Now when this explant has been, when this plant is, when this hybrid plant is there, we can take our explant from this plant. Then from this explant we can go for establishment of callus culture. First we have to make, always we have to make from explant, we have to go for establishment of callus culture. Then from callus culture we have to go for the screening of the secondary metabolites. We have to go for the screening of the secondary metabolites in the callus culture. Then this callus culture is always on the solid media. Then from the solid media slant, we have to take this callus into the cell suspension culture, so that the cells will be, due to the agitation, cell will for this the suspension will be formed as a single cell suspension culture. Then from single cell suspension culture we have to allow cells to increase their biomass. Then when they have increased their biomass, we can go for mobilization techniques to these cells. Then we can, after immobilization of these cells, we can use these cells for our secondary metabolite production. So here are the few common techniques as we come to across till now, that how immobilization is occurring. We can use absorption method, we can use encapsulation method, entrapment method, cross-linking method, and covalent method. These are the few important techniques which are used for immobilization techniques. But for plant biotechnology, it's not applicable for plant biotechnology. All the techniques are not applicable for the plant biotechnology, as the plant biotech cells are very sensitive with respect to its pH, temperature, or the composition of the media. So we had to be very specific for tissue culture cells. Now, the methods of immobilization of cells in case of plant biotechnology are: first, gel entrapment by the polymerization. Second is the gel entrapment formation by precipitation method. Third is the surface immobilization method. And the last is the immobilization by embedded method. So now the first, gel entrapment by polymerization method. In this case, what we do, we take a monomer or the mixture of monomer which is polymerized in presence of cell suspected culture. When the polymerization takes place, our cells will be entrapped in that polymer, and the commonly agent used for the polymerization is here polyacrylamide. The method is actually based on the free radical polymerization of acrylamide in the aqueous solution. This technique has been mostly applicable for the Catharanthus rosaceus and Silybum marinum. These are the two different plant species in which we were able to get secondary metabolite production by gel entrapment polymerization technique. The second technique which are used for the immobilization of plant cell is the gel entrapment formation by precipitation. So gel may be formed by precipitation. So we can use here natural polymer or synthetic polymer. These polymers can be precipitated by changing in one of the parameters of the solution. So what will be the parameters of the solution? It will be the temperature. By changing the temperature of the solution, we will get precipitate. In this precipitate, our cells will be entrapped, or we can increase or decrease the salinity, as in case of salting in and salting out technique, or we can change the pH of the solution, by which precipitation of the medium will occur and our cells will get entrapped. So several methods can be used for this entrapment method. For example, we can get precipitation by thermal treatment, like increase in the temperature. There will be some disruption to the viability of the cells, but this is one of the techniques by which we can immobilize our plant cells. Now the third is the surface immobilization technique. Third is the surface immobilization technique. In surface immobilization, we can use two different type of matrices. First is the natural matrices and another is the artificial or synthesis matrices. The example of the natural matrices is the use of callus layers or cellulose. On the cellulose surface or on the callus, we will immobilize our cells. We will be able to immobilize our cells. Or there is one example like cells of Solanum, genus Solanum, were covalently linked to the beads of polyphenylene oxidase, which has been achieved by using glutaraldehyde. So Solanum genus is cross-linked to the beads of polyphenol oxide. This surface immobilization is achieved by the use of the chemical is glutaraldehyde, and this technique is applicable for the genus Solanum. And the last is the immobilization by the embedding method. This embedding method is mostly carried out by the agaros. As you might be familiar with the things, the temperature dependent solubility of macromolecules like agaros, or we can use the solubility of sodium and calcium salt in case of alginate. These solubility are utilized for the formation of polymeric gels. Insoluble are formed These insoluble gels are formed under the cold condition, as we can see in the agar. When there is cooler in the temperature, then gel formation in case of agar takes place. And for the alginate, we have to use aqueous solution of calcium chloride. And for the alginate, we have to use aqueous solution of calcium chloride in which the gel formation takes place. It has been said that the embedding method, we don't have the uniform structures. Like here, what we do, we use pipette, parcher pipette, or dropping aperture something. From this dropping aperture, we are able to pour our drop either in the cold solution or in the calcium chloride. This dropping will decide the size of your beads or size of your drop, ये agaro's droplet. So this is also the risky method, whether we will be able to get appropriate size for the cells for immobilization or not. It depends on your droplet method, or we should be having such droplet method which should be automatic, like we should be able to get equal size of these droplets, but still they are having its limitations. But this is the most preferable method which we can use. Even we can use this immobilization of embedding method even in our labs as well. So if we will talk about the advantages of immobilization cells, there will be many advantages over the disadvantages. So first is a little or no conformational change of the enzymes. Whatever the byproducts we are getting by this immobilization of cells, it has been observed that there is little or no conformational change of the enzyme. Whatever the enzymes we are able to get by this immobilization technique can be directly used for their respective fields. Like if we are getting lipases by this technique, then we can extract this lipase, then we can use this lipase some elsewhere. This technique, the immobilization technique, is very simple. We can carry it out in our labs. They don't need specific equipments or sophisticated lab to develop this. And the components which are used for the immobilization are very cheap. So we are having a high biomass level, as we have taken a single callus. From single callus we have got the single cell suspension culture. Then these cells can be recycled again and again. Regeneration process can be occurring. By the regeneration process, we will be able to have high biomass level. As we said that there is no specific reagents required for the immobilization of cells. The reagents which are utilized for the plant tissue culture labs are very costly, as whatever the reagents we are using for tissue culture are very pure. As the purity of the product increases, the cost of the product as well as increases. And the separation of cells from the medium is easy. How? The cells are embedded, immobilized in the gel. So whatever the secondary metabolites are present, they are present in the medium. We will go for simply filtration, or by the centrifugation technique we can separate cells from the medium. Then from this medium we can go for extraction method by the downstream processing for the extraction of the secondary metabolites. The immobilization technique is a continuous process in which we are continuously recycling the cells and continuously we are using the medium as we said earlier, that these secondary metabolites production can be done in the automatic fermenters. When we are having automatic fermenters, we know that within in this time our media would be utilized by the cells. Then they will be able to produce secondary metabolites within in this time range. When this time range has occurred, when this time range has come, then we can extract our cells. We can use these cells in the next fermenter. Then we can go for the downstream processing of the secondary metabolites. There is the first formation of growth. The cells will grow first fully, then at the last the product will be formed. If you will remember, maybe I don't know, in the fifth sem of your plant physiology, we have discussed the plant growth curve, lag, log, and stationary phase. And the secondary metabolites are always formed at the stationary phase. We can say that stationary phase there is no growth. It means cells have to grow fully, then only they will be able to produce the secondary metabolite. So by using these immobilization techniques, cell will not get aggregated, or there will be no formation of foam. As we have, you might have studied this in the fermentation technology. While going for the fermentation, there is a formation of foam as well, but for the immobilization technique, we are not having formation of the foam. These cells are recyclable. We can regenerate these cells again and again. Obviously we can reuse these cells. Now everything has its disadvantage. So it's like, first we have to take care of the cell permeability. Secretion of secondary metabolites requires cellular transport, or we can say artificially altered membrane permeability. The efficiency the efficiency of the production process depends on the rate of release of product. Production process depends on the rate of release of products rather than the actual rate of biosynthesis. Whatever the secondary metabolites are formed in the cell, it should be efficiently released from the cell. But in case of immobilization process, it has been observed there is the reduce in the biosynthetic capacity of the cells, because these cells are not that permeable to excrete secondary metabolite into the medium. So efficiency is low. This process is very slow. If we will compare the overall process, overall the investment in this technique, then we can say that the efficiency is, or this is a very slow method. Deabsorption of enzyme protein result from change in the temperature, pH, or ionic strength. While we know that whatever the plant tissue culture bioreactors are there, we have to be very specific. We can't open the lid, it will get contaminated, then whole batch we have to thrown out. If there is change in temperature by the metabolic activity of this cell, then the enzyme will lose its enzymatic activity. Or if by default there will be increase in the pH or ionic strength of the medium, then the whole batch will have to discard. Now this is the we are having list of products which are till now extracted from the plant cell by use of immobilization technique. So biotransformated cells can use in agarose gel, alginate, polyurethane foam, or we can use these, and we can use these immobilized cells immobilization technique for different plant species. Like we can use agarose for Catharanthus rosaceus, and the end product of this is edgmalicine. This edgmalicine is the secondary metabolite, and it has been observed that this edgmalicine is important in lowering our blood pressure. This edgmalicine is important in lowering our blood pressure. And the another product is digoxin. Digoxin is involved in the various heart conditions. We can use this compound, this secondary metabolite, even if there is a heart failure or arterial fibrillation also. The another product which is from papiver somniferum, we from papinum somniferum we are able to extract secondary metabolite, that is codidine. This codidine is act as a painkiller. We can use this codidine when in treating the pain or coughing or diarrhea. And the most important, you might have heard chiconin. This is one of the alkaline and it is one of the best antioxidant. It's having best antioxidant property against the Staphylococcus aureus or Staphylococcus epidermidis. This chiconin is actually the dye from we are able to extract it from the plant cells. So this is the first natural colorant that has been commercially produced by the plant cells. That has been commercially produced by the plant cells. So these were some important secondary metabolites which the plant cells are able to produce.